Hola a todos, bienvenidos a mi canal. Soy Mariana y en este nuevo vídeo vamos a tener, es un vídeo bastante especial. Os explico por qué. Resulta que se trata de una entrevista que tuve el honor de hacerle al doctor Jed Fahey. Y el doctor Jed Fahey, pues él es biólogo, pero aparte, tiene un máster de la Maryland University y aparte también pues su doctorado de la Hopkins University. Pero bueno, él se acaba de jubilar se acaba de jubilar, pero ha estado de casi 30 años de profesor titular en la Hopkins School of Medicine y ha dirigido, es el director del Cancer Chemoprevention Center asociado a la Hopkins. Os comento, él es uno de los mayores expertos a nivel mundial en el estudio de los fitoquímicos presentes en las plantas, particularmente verduras crucíferas y su acción protectora frente a la salud y sus mecanismos de acción. Resulta que él está muy enfocado, como os he dicho, uno de los mayores expertos mundiales en brócoli, verduras crucíferas en general y brotes de brócoli, y como pues el sulforafano del brócoli actúa a nivel de laboratorio. También han llevado algunos ensayos clínicos. Es muy interesante cómo actúan estos compuestos presentes en las verduras crucíferas y he tenido el honor de entrevistar a Jed Fahey y al doctor Jed Fahey y bueno, pues era para dejaros la entrevista porque voy a abrir una nueva sección en el canal dedicada pues eso, a entrevistas de científicos e investigadores que vaya conociendo, y creo que es muy interesante porque las verduras crucíferas es un tema que vamos a tocar más a fondo en el canal. Estoy preparando varios vídeos al respecto que voy a ir publicando sobre la evidencia científica sobre el consumo de verduras crucíferas, como su mecanismo de acción. Y bueno, para empezar esta serie de vídeos pues os dejo aquí la entrevista. He puesto subtítulos en español y bueno, pues es larga pero la podéis ver a trocitos y espero que os guste un montón, amigos. Un beso y no olvidéis en darle al me gusta si os gusta el vídeo. Adiós.
Well, let's start with the interview that I have prepared. Well, the first question I have is that, first of all, it would be interesting to talk about, in a general manner, about the benefits of cruciferous vegetables. I mean, the diseases they could reduce the risk of, and what's the level of evidence, and the compounds that are responsible for those effects.
I'm sorry, the compounds what? Or
that are responsible for those protective effects.
Yeah, so we've been studying the cruciferous vegetables, in particular broccoli, for a long time now, for over a quarter century. But my mentor, Paul Talalay, discovered with Yushin Zhang, his student at the time, discovered sulforafane in broccoli. And they discovered it they found it at very low levels. And then I went on to find it in broccoli sprouts. We realized that it was the precursor of sulforafane that was present, glucoraphanin, that gets converted to sulforafane when you chew a plant or when an insect or a predator chews it. And after all, those are their defense compounds. Our calculations a long time ago were that to get a reasonable dose of sulforafane that might be biologically active in human beings, you would need to eat something like 1.25 pounds of broccoli to get a similar dose of sulforafane as you would get from eating a couple of ounces of broccoli sprouts. And after all these years, we still don't have a great idea of proper human, quote, dosing. So remember, it's not a drug, it's a natural compound, and it's part of the whole matrix of fibers and many, many thousands of phytochemicals in the plant. And as such, the minute we start talking about, quote, the dose, we're we almost by definition have in mind treating a specific disease as if we were treating with a single drug. So I think it can be a little bit dangerous to go down that path. But that said, when we try to compare what's available in, you know, a pound of broccoli or a couple of ounces of broccoli sprouts or a very small amount of broccoli seeds, which can be extracted and produce a supplement, dietary supplement, then we almost always wind up relating it to the dose required to cure or treat a condition in an animal study or a human study. So,
So,
well, so I
yeah.
was just going to say, with that in, you know, with that in mind, we and others have looked at a whole bunch of really disease states or conditions or syndromes and looked at the ability of sulforafane to affect them and gotten positive results. And obviously, well, not obviously, but we were very careful, and others have been very careful about quantitating and measuring the amount of sulforafane or its precursor that was delivered to those people. I'm very hesitant to talk like this, but I do all the time. But I'm hesitant to talk about it because if this natural compound inches too close to being called or thought of as a drug, then I'm afraid that I find it hard to imagine that the regulatory agencies, like in the US, it's the FDA or EFSA in Europe, that they would try to classify a head of broccoli or a bowl of broccoli sprouts as a drug. But essentially that's, you know, it's food as medicine, right? It's food as drugs. So it's a fine line that we have to be a little bit careful with when we talk about it. And so I really don't like talking about a dose of broccoli or a dose of broccoli sprouts. But the reality is that we can't help ourselves but equating the amount in the plant to the amount that you get. But just we have to remember, all of us, that when we're eating a plant, we're getting a whole bunch of things together. And scientifically, at this point, we're still not smart enough to be able to do the assessment of how, you know, 100 compounds interact together in the human body. Anyway, that's a long answer to a
Yeah.
simple sounding question.
That's a very interesting point because many people think that there are like magical foods, and it's not that way. If we want to reduce the risk of chronic disease, the key is to follow a healthy dietary pattern with a whole foods plant-based diet, and obviously cruciferous vegetables are going to be protective with lots of mechanisms, and the other vegetables also, and legumes and whole grains and all this stuff. But we cannot, when it comes to reducing the risk of chronic disease, it's not about one compound. It's the interaction of all those and the effect that the healthy lifestyle has on your health.
Absolutely. And you're familiar, I'm sure, with blue zones.
Oh yeah, it's amazing. Yeah.
Yeah, and so I mean that is perhaps not as scientific. It's a lot of survey research, but so many places now, so many studies have shown that connectiveness, positive attitude, and exercise, you know, go hand in hand with
Yes,
diet for sure.
absolutely.
Yeah, yeah.
It's like the we have it's like a box full of things, and you have to change your lifestyle in
Yep.
a complete way. It's
Yep.
not many people think that by eating, I don't know, kale or blueberries, they are gonna compensate all the alcohol they have consumed the night before. So we have to keep in mind that we have to follow like a healthy lifestyle, healthy dietary habits,
Yep.
and change the way we live. Yeah.
Yep. Yeah, certainly. I mean, you're as a physician in training, you're very well aware that you can't sort of have a cigarette and then offset it with a bowl of broccoli. I mean, you can't live like that. But that brings me to a point that I'd like to stress. I mentioned you're a physician in training. One of the problems that here in this country, and I think in most of the world, we see in physician training young students to be doctors, is that there's very little appreciation for food as medicine, for cooking, for, you know, if you're a physician and you're at least up on your nutritional literature a little bit, and you recommend to a patient that, say, is overweight and needs to lose weight or something, you maybe recommend that they switch to a more plant-based or plant-forward diet. So they look at you and they say, yeah, okay, thanks, doctor. How do I do that? And so often you see doctors in this country just sort of go blank, you know, well, eat more. And, you know, the specific recommendations are just, and the ways to prepare these foods, they punt on. And so you have to.
I have like several videos in my YouTube channels where I try to explain how to cook vegetables, for example, broccoli, so as to get all this sulforafano, chopping it before, so that the myrosinase can get into contact with the glucosinolatos and get the sulfur. Oh, that's like amazing. I want to teach people how to, you know, I want to inform them, and also how can they cook and prepare all these foods so as to get the most benefits, and yeah, and it's very easy. Once you know it, once you learn it. For example, I have been on a plant-based diet well, I'm on a plant-based diet since I was 14 years old. Mm-hmm. So for me, it's like natural, you know, like cooking all these things, and it's like very easy for me, because I decided to do all this change because I had a lymphoma. Like when I was 12 years old, I had a lymphoma, and my mom at that time, she had a breast cancer. So I started to do my own research on how diet and lifestyle can influence our risk of cancer and how can we prevent it. So I changed my lifestyle completely because I wanted to take action. I wanted to be, I wanted to do something for my health. And I knew that, of course, medicine and all the treatments are very effective and so on, but you can improve and you can reduce your risk a lot also with healthy diet and a healthy lifestyle in general.
Absolutely, and I think I think it actually makes a more important space all the time in healthcare for dietitians and nutritionists to have an impact. I wish their services were better compensated by insurance programs. Certainly in this country, they're not. Other places of the world, I believe, are a little bit more accommodating to them. But, you know, and there are some really good programs now for to teach physicians about food as medicine. There's one that's, I think, franchised by Tulane University here in the U.S., and there are a number. I have friends who are instituting these programs in medical schools. For example, at the University of Maryland, where I used to teach, Chris D'Adamo in the Institute for Integrative Medicine there has such a course. So they're catching on, and there are very good people that are doing them, but it's still not nearly enough.
Yeah.
And, you know, you and I we're talking about the microbiome before we got on the air here. And I think one of the things that one has to really look to over the next 5 or 10 years is a much better understanding of how eating the whole plant. I mean, let's just take broccoli for an example. Eating the whole plant versus just taking a concentrated dose of sulforaphane, how that affects the gut microbiome. I know it will, I know it does, but I can't tell you how. You know.
Because all the fibers and all the different bioactive compounds that are present in these vegetables, we don't know how they interact, how they affect our microbiota, but the thing is that it's good for us. So
right. And we know
we don't know, but it's good.
And we know, for example, that, I mean, if you just take that comparison, if you gave a single dose of pure, let's just say sulforaphane, it would probably have a selective antibiotic effect on some of the critters in your gut. If you gave it in the presence of all of the fibers and proteins and so on in, you know, a pound of broccoli or a meal rich in broccoli, you would be nourishing your gut microbiota and perhaps even directing them to grow in a certain way, because you'd be providing fiber for them to break down and to act on. So it's even though the microbiome science is, you know, 20, it's a good 20 years old now. It's still the Wild West in a lot of ways. There's so much we still don't know. And there are a lot of quacks out there waving their hands and giving tests and trying to make pronouncements about what this means and what that means. You have to be careful interpreting advice on things you can do to change your microbiome. Yeah. But it's coming. Yeah.
So the other question I wanted to ask is like, we know that myrosinase is the enzyme that is responsible for this conversion of glucosinolates into isocyanates, but how does it work, and what's the role of gut microbiota in all this?
So the way it works, let me see if I can pop a slide up here for you, because that's probably the easiest way to do it. The easiest way to share it with you. There we go. I have one here. I have to just screen share. Here we go. Okay, can you see the slide now?
Yeah.
So, I mean, I've shown this slide before, and it's been published, or something very much like it has been published many times in many places, actually. But so you have this precursor in the plant. It's water soluble and heat stable and not very reactive at all. It doesn't do much biologically. It used to be called a storage form of sulfur, because you see there are actually 3 sulfur molecule atoms in it. In the plant, there is, in the same cells that have glucoraphanin in a little vacuole, so a little liquid bag, if you will, there's this enzyme called myrosinase, which best we know is membrane bound. So it's tied to certain structures in the plant cell. When an insect or a predator comes and chews on the plant, it breaks open the bag, releases glucoraphanin or other glucosinolates, and they get converted enzymatically by this enzyme myrosinase, which acts very quickly, and it cleaves off a glucose, and then the molecule undergoes a rearrangement spontaneously, non-enzymatically, and it produces the isothiocyanate, characterized by the N double bond C double bond S group. So basically, it's chopping this piece of the molecule off, what you see on the left side here, and there's a rearrangement. The sugar, the glucose goes away. Well, it's released. This molecule is it's not as water soluble. It's heat sensitive, it's reactive, it's a lacrimator. It makes your nose run and your eyes weep if it's very potent, as it would be in cruciferous vegetables like wasabi, maybe, or nasturtium, or mustard, or radish, where there's a similar compound to this formed, but not quite as potent. So if you cook broccoli, let's say, or anything, what happens to an enzyme? An enzyme is denatured. It goes away. Well, it doesn't go away, but it's denatured and it's no longer active. And in that case, you have to count on enzymes with myrosinase activity in your gut microflora. And interestingly, all of us have these enzymes. All of us have microflora with these enzymes. We've looked at hundreds, and others have looked at hundreds and hundreds of people, and they've asked the question by giving a dose of glucosinolate and monitoring for the isothiocyanate like sulforafane or its metabolites in the blood or urine. Everybody's got this reaction. And we were actually able, over 20 years ago, we did a study where we had people self-administer fleet enemas to clean out their lower GI tract, reduce the bacterial population dramatically. And then we gave them a large dose of preoperative antibiotics to further reduce the bacterial population in their guts. And then we challenged them with glucosinolate bolus. And lo and behold, when we followed them, they didn't produce any metabolites. And over 2 or 3 weeks, their ability to produce them rebounded as their guts repopulated. I stress this was done, it was actually about 25 years ago. And this is not an experiment I would ever do today to someone, knowing what we now know about, first of all, about the long-term effects of antibiotics. And second of all, just realizing the harm that can be done to the gut microflora by wiping them out. Sometimes you have to do this pre-surgically, but as an elective, low-risk procedure, certainly our institutional review board approved it, and this was a fine study to do back then. But as with all things, your understanding evolves, and I wouldn't do this study nowadays. Anyway, that we were able to show that indeed the ability to do this conversion was mediated by gut microflora. And just one final thing, now in the days of microbiome science, certainly for many years now, people have sequenced bacteria in the gut and know some of them that have myrosinase-like activity. So that's all much better known now. Sorry.
Yeah, that's very interesting because in this part, we see that how our microbiota is, that's gonna play a crucial role in the way we get benefit from sulforafane, or if we can actually get it. Because I think I'm of the opinion that if you follow a healthy dietary pattern that includes lots of veggies and plant-based foods, and particularly cruciferous vegetables, you are gonna have these microbes that are able to produce these myrosinase, and in that way get the sulforafane. So as we have talked before, it's we have to change our diet, and also I think that it would be particularly interesting to see the impact of some fermented foods in the gut microbiota, and how they can improve its diversity, and in that way we are like we can get more benefits from the vegetables, like the case of cruciferous vegetables, especially broccoli.
Mm-hmm. Mm-hmm. I agree. I think that's a statement, not a question, right?
Yeah.
Yeah.
And one thing, I cannot see you. I don't know if there is a problem with the video, but
You can't see. What are you seeing?
I cannot see you. No, I listen to you, but I cannot see your
Oh.
face.
No, I'm seeing both of us, so I'm not sure what's going on.
Well, I think everything is being recorded okay, so I suppose there's no
Well,
problem.
it's being recorded, but hopefully the video.
Okay.
I know. Go to your, I think probably the top right of your screen. There's something for view, and
Okay, yeah.
make sure it's on gallery view or maybe speaker view. Change
Okay.
the setting there.
Yeah, I have changed it. It's much better, but I cannot see your face. I don't know what's going on.
I don't know. It must be a bandwidth thing. Let me share screen again for a second
Okay.
and then stop it. Okay. Do you see both of us now?
Yeah.
Oh,
I see the presentation, how these
do you see both of us though? Do you see both of us in a little postage stamp size window?
I can see myself, but not your face.
How about now?
No, I don't know what's going on, but I don't know if it's I don't know, but I think if you are seeing me and everything is being recorded okay, I don't think there's any problem.
Okay, yeah, I mean, so it's being recorded on this end, and I believe what's being recorded is what I'm seeing, but yeah. Well, I don't, we don't have a huge amount of time anyway. We can always redo it.
Your camera working.
What's that?
Your camera is working okay.
Yeah, I mean, I'm seeing both of us on this. Yeah.
Okay, no problem. No problem. So let's continue with the interview. So
Okay.
let's talk a little bit about what you are particularly focused on broccoli and moringa. So when it comes to sulforafane present in broccoli and broccoli sprouts, which are the we know how they work. You have studied a lot the NF2 activation, the FOXO pathway, also the phase 1 enzymes, phase 2 enzymes, and also I know that they work by inhibiting the degradation of P53 gene. But when it comes to cancer, for example, which studies you have carried out that, and which is the preclinical and clinical evidence about broccoli and cancer? What have you seen?
Well, I think a lot of the evidence is more on sulforafane and carcinogenesis because, as you know, it's very difficult to do interventions with people who have a cancer with a food. It's difficult for, or even with a compound, a natural compound. It's difficult for a lot of reasons. First of all, if you're going to intervene with a food, a drug company is not going to spend the millions of dollars on the trial to sponsor it, and the funding agencies don't do a heck of a lot of it, frankly. Secondly, if you're doing an intervention to try to treat or prevent, or to try to prevent cancer, you have to look at a large number of people who might get that cancer, right? So that means look at a high risk group, but still, it's a very long, very multi-year trial with a lot of people, and you have to monitor a lot of endpoints, but in particular, of course, cancer. So those trials are obscenely expensive, and I don't think anybody has actually done one because of that. It would take an investigator's lifetime, really, to do one trial properly. in terms of therapy, once someone has cancer, there are some people who will treat naturally. Most of those aren't involved in a clinical trial, especially if the standard of care is to give drug X. It's very difficult to get a clinical trial together to say, okay, this arm will use, cisplatin, and this arm will eat broccoli. I believe most ethical review boards won't go for that, and probably with good reason. So the answer to your question is a lot of the data we have comes from carcinogenesis models, animal models, or cell culture. And in that vein, let me screen share with you. And can you see this?
Yeah.
Okay. So I put this chart together, and Tom Kensler and I published it in a paper we did just a few months ago, actually, in Frontiers in Nutrition. As you mentioned, the NRF2 KEAP1 pathway is quite central. It's central in that sulforafane is the most potent naturally occurring inducer of the NRF2 response that we know of. Certainly there are synthetic drugs that are more potent inducers, but they have side effects. But over the years, over the last 25 to almost 30 years, many other mechanisms, some of them are shown in white here, have been uncovered. I did work on Helicobacter infection.
Yeah,
There's
very interesting.
There's a paper just out a few months ago on the anti-COVID activity in vitro and in an animal model of sulforafane. There are various papers on antibiosis. So there's clearly some selective antiviral and antibiotic activity. You mentioned the antioxidant response. That's indirect because you're with primarily NRF2, you're inducing antioxidant enzymes. Sulforafane itself is not a direct or a self-sacrificial antioxidant. And then there's anti-inflammatory activity and immune response activity. These dotted lines mean that there is some evidence, but it's certainly not ironclad. And for all of these, there is animal evidence, animal model evidence, and in some cases, some clinical evidence. For example, in the neurodegenerative, neurodevelopmental realm, we've done some work with autism. We see effects.
Tell me a little bit about the clinical evidence that you have.
Let me come back to that in a second. But you asked specifically about cancer. Didn't mean to do that. So there's evidence that sulforafane is effective in inhibiting tumor initiation, promotion, progression, and metastases, all of them really in different models. And these, because it's too many to fit in this picture, this is a list of just some, and this is in our paper, just some of the pathways that other people, this is mostly other people, not us, have determined are part of the modes of action of in the prevention of carcinogenesis. So you asked about cancer specifically and mechanisms. That's sort of the 30,000-foot view of what happens there. I'm sorry, you wanted then to talk about the autism work?
Well, so before that, I would like to say that we can see that sulforafane acts in very different ways, and it can induce the activation of our immune system, anti-inflammatory effects, antibiotic, antiviral, anti-cancer, and lots of and also it could have like a it could exert neuroprotective effects, but mostly all this is preclinical evidence, and the clinical evidence is limited. But I think that when it comes to cancer, it's important to highlight the fact that with cruciferous vegetables, we have epidemiological evidence that shows that people who consume cruciferous vegetables, including broccoli and cauliflower and all this stuff, have like a reduced risk of several types of cancer. And this is so we could say that when it comes to cruciferous vegetables, the grade of evidence is more or less good, because we have like epidemiological studies, some clinical trials, and also we have some preclinical research. So I think that maybe the sulforafane has like preclinical evidence mostly, but cruciferous vegetables in general, I think that they are a good an excellent component of a healthy diet. Yeah.
I'm very sorry that I neglected to mention that. You know, if I didn't know better, I would say that you and I rehearsed this with each of us feeding the other setup lines. But absolutely, what you say is absolutely true. Many times when I give lectures about our work, I lead off talking about the epidemiologic evidence. I didn't have a slide here to share on it, but you're 100% right. And in fact, it was the epidemiologic evidence that started in 1975, 1978, a hell of a long time ago, which really started moving this field of, highlighting cruciferous vegetables as potentially cancer preventive. There was work that a guy named Graham did, a researcher, very respected researcher by the name of Graham, did in, I believe it was 1975 or 78, showing that people who, he divided, as I recall, divided cruciferous vegetable consumption in a fairly large population sample size into quintiles or quartiles, or I believe it was quintiles. So those who consumed most on a regular basis based on food frequency questionnaires versus those who consumed least, and found a pronounced effect, a protective effect on colorectal cancer. And those studies persisted through the years, where people looked at prostate cancer, breast cancer. So the ones in which broccoli or cruciferous vegetables were particularly protective were breast cancer, prostate cancer, colorectal cancer. I believe those are the three that come to mind now. There are a few others, and that epidemiologic evidence gave us courage or ammunition, shall we say, to do what we could do as basic scientists, which is look at specific compounds which mechanistically looked like they were working in the same, to support the same disease preventive capacity as the vegetables themselves. But by now, there's a body of, hundreds and hundreds of epidemiologic studies that support the fact that cruciferous vegetables are protective against cancer and a bunch of other chronic diseases. So you're absolutely right. And it's very important to come back to those studies and to not move too far ahead with the mechanistic stuff and the very specific animal models with a single compound without tying it back to what the epidemiology says. I mean, you can't prove causation with observational studies, but it certainly strengthens the case of it has to go hand in hand with the basic science, the basic research on mechanisms, right?
Yeah, I think that we have to, when talking about diet and how a particular a dietary pattern or a particular food it's good or not for our health, we have to take into consideration lots of things, and not just the clinical evidence, also epidemiological, and also clinical trials, and when all these components go in the same direction, is when we can say, well, that's good. But we have to it's necessary to have all these components, because if not, for example, right now there are like lots of trends when it comes to diet, like the ketogenic diet or the paleo diet or the, I don't know, many diets, but we don't have like the enough evidence for that, because we maybe have a handful of clinical trials, but we don't have that epidemiological evidence and those and lots of clinical trials. And for example, when following a plant-based diet, we have like a higher grade of evidence. So that's the thing.
Yep. Well, I agree.
And I'm particularly interested in the antiviral and antibiotic effect of sulforafane.
Yep. I'm not going to say much about them because I don't want to raise false hopes, but we've certainly published our work on Helicobacter, and it's quite selectively antibiotic in vitro. We are able to reduce colonization in an animal model, and in the clinic it wasn't as impressive, frankly. But having said that, I've always felt, and this is a source of some debate within the gastrointestinal gastroenterology community, I've always felt that if someone has a severe Helicobacter infection, and you can, or has an infection rather, and you can keep the bacterial titer down, keep the number of Helicobacter down to a low level, so that they don't cause ulcers, and which can eventually lead to stomach cancer. That's helpful. And there are many now anecdotes from people, they're just anecdotes, but where people have not tolerated the quadruple therapy or triple therapy that's typically given for Helicobacter infection, and the metronidazole and clarithromycin and various combinations of antibiotics are given, and there are many people who can't tolerate it. It's apparently a pretty bruising regime. As well, there are people who are treated and still don't resolve their Helicobacter infection. So anyway, a subset of that population has self-treated with broccoli sprouts, or in fact, cranberry is another food that's been shown to be quite effective in vitro and in animal studies. And so, you can reduce the titer, although you may not kill every last Helicobacter cell in your body, but you can keep them at bay with a simple dietary approach. So that's part of the antibacterial. In terms of the antiviral effects, I mentioned in BioRxiv, there is a paper that was posted in March by a group at Hopkins, friends of mine. Ordonez is the first author, and it is not peer reviewed. I mean, it's undergoing peer review somewhere, I believe, but they present, in my mind, some pretty impressive experiments in test tubes or Petri plates, if you will, and in mice, looking at the effects of sulforafane on COVID-19 or SARS-CoV-19, SARS-CoV-2, sorry, and another coronavirus. There are papers that have been subject to peer review that have been published speculating about the ability of NRF2 inducers like sulforafane to combat SARS-CoV-2. So people are certainly thinking about it. I don't want to raise false hope. There's been enough of that raised all over the world by politicians and others. But I mean, there's no question that everybody's searching for therapies, right? I mean, for approaches to reduce the impact of COVID-19 and to reduce the impact and severity of infection once people contract it, if they do. So I think, you have to keep your eye on the scientific literature for further progress, further results, but obviously many natural compounds are being examined. And I don't pretend to be an expert, and certainly neither you nor I are suggesting that anybody try any of these things. But right, I hope neither of us are. But
And this is very interesting, a very interesting area. And I think that, of course, we have to say the truth and the evidence that we have, and we have mostly preclinical evidence. But one thing that has happened to my mom is that she, for several years, she has had herpes virus. And since she has broccoli every single day, it has disappeared.
Mhm.
So I think that I don't really like to talk about these things, because I don't really like to say things that are not scientifically proven, or they have a, but
Right.
that's true.
Well, I think, many of us that do basic research are taught, well, I think we're all taught to not pay attention to single anecdotes, but on the other hand, it really is those anecdotes that get you thinking about where to go next and what connections might be. So I like to hear those kinds of anecdotes, because many anecdotes combined really starts to get intriguing if they all go in the same direction. That said, we all need data, though. So we're getting a little bit short on time. You had said something about Moringa. You had asked me about
I
that.
wanted to ask you several things.
Yeah.
Several things. Okay. So moringa, it's glucomoringin, which is like is moringin more effective than sulforafane, or more powerful than sulforafane? What do you think? Is moringa better than broccoli, or what do you think?
Interesting question, long answer, of course. But so better is a relative term. So if you isolate and purify those two compounds and run them in a head-to-head test in various assays, sometimes moringa wins, sometimes sulforafane wins. So the precursor is called glucomoringin. The isothiocyanate is called moringin with an N at the end. So it depends on the assay. In terms of efficacy and potency against Helicobacter, for example, they're equal. They're both the same. In terms of inducing some protective enzymes, sometimes sulforafane, in some assays rather, sulforafane's better, in some moringin is better, which I'm sure relates to their efficiency in binding at certain sites, getting into cells, and so on. So the best answer is they're about the same. But then that leads to the food related question. And if you say, should I be eating moringa or broccoli? That's easy for me to answer. And my answer is, it depends on where you live. So in the temperate zone where I live, and I believe Granada is subtropical, right? Where you are?
Yeah.
Yeah.
Here lots of broccoli is grown. I have a moringa, so we
Yeah.
have everything.
So you have everything, but a lot of people don't. So in the cooler temperate
Yeah,
zone, in the cooler temperate zones, broccoli is widely available, and that's that's what you should eat in terms of carbon footprint and food miles and all of that stuff. But on the other hand, although it's a little bit more expensive, moringa, dried moringa, is widely available now in the United States and I think in many places in Europe. And full disclosure, I'm actually a scientific advisor for a company called that sells moringa dried moringa leaf powder in the U.S. They source it responsibly in Africa from primarily women's collectives, farms, and so it is fine. I mean, it's equally good to take as broccoli, but of course it's dried. So
here in Spain, you find it as you have told us.
I'm sorry, I missed.
I consume it's right. It's a leaf that the plant is grown here in Granada, but they sell it dried. But the another question I have for you is how to integrate moringa powder into your daily basis, because for me it's a little bit hard. I added it to the salad the other day and I didn't like it so much. So how do you consume moringa?
Okay, so let me finish the previous answer to the previous question first. I'll come right back
Sorry.
to this. No. But I'll come right back to it to this question. But so if you live in the tropics, chances are you're not going to have that good access to cruciferous vegetables, or you might, but you probably will have access to fresh moringa leaves, and then it's I certainly would recommend it. A lot of fiber, a lot of vitamins, and very high protein, which is somewhat unusual for a leafy green plant. So you asked how you consume it. So if you have dried moringa leaves or dried moringa leaf powder, one way to consume it, and the way that I most frequently consume it here in the temperate zone where I have my bottles of moringa leaf powder, is to make a tea out of it, or an infusion. And it's interesting. We published a paper a couple of years ago. It's on my website, jedfahey.com, where I have links to publications. If you make a typical herbal tea with boiling water, a hot water infusion of moringa leaf powder, you extract the glucomoringin, the glucosinolate, and it does taste strong. There's no question about it, the tea. If instead you use cold water or even ice cold water, you just put it in the refrigerator and steep it for a couple of days. What you do is you activate the myrosinase in that leaf powder, as long as it's fresh leaf powder that's been stored well. And this is where a company like has advantages in terms of being a supplier. So you wind up converting glucomoringin to moringin in the water, in the leachate or the tea. And actually that tea has a sweeter, less astringent taste than the hot tea. So I don't have it right this minute, or I'd show it to you, but I frequently have a pitcher of moringa tea in my refrigerator that I drink during the day in the summer. You can certainly...
That's the way I'm gonna prepare it, yeah.
Yeah, and you can certainly use moringa in all sorts of recipes, but it's more conducive to putting it in recipes with sort of strong tastes. Certainly a lot of Indian cooking it complements very well. I just was sent an email yesterday from a former student who's at UC Davis, University of California at Davis in the US. And she told me that their CSA, their community supported agriculture, which every week sends them out or gives them a bunch of vegetables that were grown locally, they're providing fresh moringa leaves there now. So, and everybody that distributes it or sells it has recipes that they suggest. So you can really incorporate it into just about anything, and just have to experiment with taste because it is harsh, as you said.
And another thing I wanted to ask you is broccoli. How do you consume broccoli? And it's a good. It's better to chop it and before you chop it, you wait for 1 hour more or less, and in that way the myrosinase get into contact with the glucosinolatos. Is that a good technique, or what's the best way of consuming broccoli?
The best way all depends on your patience level, your level of patience, not patience, but patience, and what you prefer. I like my broccoli lightly cooked or steamed. I know that will destroy the myrosinase, but I'm counting on a relatively healthy gut microbiome. And so my wife and I have broccoli at least a couple of times a week. I eat other cruciferous vegetables all the time. I actually am picking nasturtium leaves out of the garden in the back almost daily to have with my salad, but that's only during that height of the summer. And I grow broccoli sprouts regularly, and I eat them. I don't, there are various people online I know who say, well, you have to homogenize and drink it in a smoothie. I'm an old guy. I didn't grow up drinking smoothies. When I was a kid, smoothies weren't invented, So although I do enjoy them once in a while, I also enjoy just chewing on vegetables. So I eat fresh broccoli sprouts, typically put them on salad. Because I'm especially interested in making sure that I keep my antioxidant system upregulated and my immune response system in good shape. I have been for many years now also taking a dietary supplement there that contains a broccoli seed extract. And I happen to know a lot about which ones are good and which ones aren't because my elder colleague and I, Paul Talley, and I started a company 25 years ago called Brassica Protection Products. And we actually started the company to have co-packers produce broccoli sprouts. But for various reasons, the company stopped producing broccoli sprouts and now focuses on making a broccoli seed extract. And that extract is sold they don't sell it directly to consumers, but they sell it to dietary supplement companies who incorporate it into various dietary supplements. So I've tested a number of those dietary supplements in my lab before I retired, and I know which ones are reliably good and which ones are garbage. And because I think there are over 100 dietary supplements that advertise that they contain sulforafane. So I take some of the better ones that I know have the product. It's called TrueBroc from BPP, or Brassica Protection Products. So I have been taking that as an insurance policy, I guess you could say, but it doesn't stop me from eating the cruciferous vegetables and the moringa that I love.
Yeah, what I do is to steam broccoli. I normally steam broccoli.
Yep.
And I consume it with some extra virgin olive oil and all these things. And I think that by steaming it, you take lots of benefits from it.
I'm sorry, I missed that. By steaming what?
That I think that steaming is one of the best methods for
Yes.
cooking it or for consuming it. I think that you can get in that way some sulforafane, although I know that the myrosinase is gonna be partly destroyed, but I think that that it's a good cooking method. What do you think?
I agree. And so there's a subtle difference between steaming and microwaving lightly. I think when you microwave, there's less, we used to call it pot liquor. There's less liquid in the bottom of the pot. Plus, you can recapture all that. The glucosinolates are going to run out along with vitamin C. Well, vitamin C will be destroyed by cooking probably, but the glucosinolates will run out into that water in the bottom of the pot. Some of them will. So microwaving probably saves a few more of them for you to ingest. If you steam, if you steam over a bed of boiling water, usually whatever goes into that water, you don't eat unless you're making a soup or something in which you can use that for a broth. But that's the only, and many people nowadays have microwaves in the richer Western countries anyway. So yeah, either method I think is good. Yeah. And most people prefer eating broccoli cooked. I mean, if you eat it as a crudity in a plate, as an appetizer, can eat some, but most people don't like to eat, hundreds of grams of raw broccoli, and that's understandable.
Yeah, because for example, in my case, when I have like raw broccoli, I don't like. I like it a lot when I consume it steam. Yeah,
Yeah.
when it's steam, it's like great.
Yeah.
And another thing I wanted to tell you is that lots of beauty products are being made here in my region with moringa, soaps, and it's being like integrated into cosmetics with dieting. And there are several papers that show that, well, preclinical studies, but in which you can see how maybe moringa could have a protective effect on the skin. But well, the evidence is very limited. But it's interesting that that it's being incorporated in many things. Yeah.
Yes, yeah, absolutely. And I think a lot of the skin products use moringa seed oil, which has very high concentrations of the isothiocyanate or the glucosinolate, moringan and glucomoringan. Moringa oil, called Ben oil in some places, is a very suitable oil for putting on the skin, and it's very high quality. It can be used for cooking. It was even used in place of sperm whale oil in lamps for illuminating many centuries ago. So yeah, the seed oil is very, very high quality. So we have to wrap up. Do you have a last question?
The last thing I want to tell you is that I think that it's very important to talk about moringa and broccoli. I want to promote it and with scientific evidence. So I really want to keep in contact with you so as to tell you the adventures that I'm having with all this and the plantation I'm gonna visit. So I really want to keep in contact with you because as I as I have told you, you are one of my heroes, and I'd like to be one day like you. and it has been like a pleasure. It has been a pleasure to be talking with you and yeah, that's it. And I really wanna keep in contact with you. Yes.
Well, thank you very much. You're too kind, but I appreciate the kind things you've said. And yes, I'd love to stay in contact. And good luck to you in trying to help people be healthier and eat healthier, because it's critically important.
Yeah, absolutely.
More so than ever nowadays. Good luck to you in your studies, Mariana.